Hello there, I'm Jeff Lindeman and welcome to this week's VCCC Breast Cancer Grand Round discussion. So as I mentioned, I'm Jeff Lindeman from the WeHi Peter Mac in Royal Melbourne. As many of you all know, this is a fortnightly event which showcasts a lot of the latest developments in breast cancer, and we're lucky, I think, in Victoria to have a lot of national and globally leading research that is being presented, including today's presentation. I'd like to acknowledge the original custodians of the lands which we find ourselves on today, wherever that is. I'm on the lands of the Wurundjeri people and would like to pay my respect to their elders, past, present and emerging, and also extend respect to other Indigenous Australians present online. So important housekeeping issues. Of course, people are going to be very welcome to ask questions today of Shom's presentation, and if you could do that through the Q&A function, that would be preferred. You can send chats and things, but I'll try to be reading from the Q&A function, so questions through that, please. So moving to today's talk, it's a great pleasure to introduce Shom Goel, who'll be talking about understanding the impact of cell cycle inhibition in breast cancer. So Shom's a clinician-scientist at the Peter Mac in Melbourne. He recently returned to Australia, having completed postdoctoral studies at the Dana-Farber Cancer Institute in Boston for about a decade. His research is really focused on the intersections between the cell cycle, epigenetics and tumour immunology, as we'll hear today. And he's contributed to a lot of very important transgenic mouse models to study this, and has had publications in high-impact journals, including Nature and Cancer Cell. And we're going to hear, I think, about some more exciting work that's coming up in Nature Cancer. So I'll hand over to Shom now. Thanks very much, Shom, for presenting.
Thank you very much, Jeff. I'm just going to share my screen. All right. I'm hoping that's good. So, good afternoon, everyone. Thanks again, Jeff, for that very kind introduction. I think it's about a year since I gave the VCCC Breast Cancer Grand Rounds last time. And I was just reflecting before I started today about how different the format is. Last year, I was doing it in the Level 7 lecture theatre in the VCCC building. And now here we are. I assume many of you are sitting at home, as am I. And yeah, so how different that format is and how different our lives are at the end of this year compared to last year. So I just wanted to take this opportunity now, in case I run out of time later, to wish everyone in Victoria the best for the rest of this year. And it's been a very tough year, and hoping that we all can have a much better 2021. So these are the disclosures that I have. All of this is directly related to work on CDK4/6 inhibitors, which are the drugs that I'll be talking about today. Okay, so my presentation today is really going to be broken up into two parts. First of all, I'm going to talk to you a little bit about a family of drugs that we often use to treat breast cancer called the CDK4/6 inhibitors. This is really a very heavy focus for my lab. And I'll explain a lot about those, why we have them, why they're good, etc. And then the second part of my talk, I'm going to present to you some of the work that we've done over the years, exploring the effects of CDK4/6 inhibitors in breast cancer, particularly focusing on some of the less expected effects of the drugs. So I thought I'd begin by just being a little bit philosophical. You know, obviously cancers, if left to their own devices, would like to grow. This is a fundamental part of cancer, and it's a fundamental part of why cancer is such a troublesome disease. And what we know, of course, is that when a cancer is growing in size, that's really reflecting that there is an increasing number of tumor cells in that cancer as time goes on. And this is happening due to the process of cell division. So one cancer cell becoming 2, becoming 4, becoming 8, etc., etc. And in my lab, we're really very focused on understanding the pathways that drive this division process in cancer cells and also to study the drugs that we currently have available that are designed to stop that process of cell division in cancer. And I think it's worth noting that this process of sustained proliferation of cancer cells is so important. It's been nominated as one of the 6 original hallmarks of cancer in this seminal manuscript from Hanahan and Weinberg back in the year 2000. So I want to talk a little bit about cell division, this process I've just mentioned. Cell division is not a process that's just restricted to cancer. We all have cells dividing in our bodies right now, which are not turning into tumors. And so this process is not something that cancer invented. It's a normal process which has been leveraged by cancer. So what I'm trying to show you in this picture is what it takes for a cell to divide. And it really is, when you get into the details, a very complex process and a very fascinating process. So what I'm showing you here in this simple picture is a single cell which is dividing into 2 daughter cells. And in order for that to happen, a number of things have to take place. Most notably, the parent cell needs to replicate all of its contents, its DNA, as is shown here in the middle of the cell, as well as all its other contents. Those contents then need to be separated into 2 distinct sides before the 2 daughter cells can then bud off and separate. This is a very, very tightly controlled process in the normal human body. And when I think about cell division as a normal process, I think one analogy or one example that we can think about is the process of wound healing. So you can imagine here that if you cut yourself and you have this defect in your skin, your body immediately goes into the process of trying to heal it. And the way that works is that there are signals generated in this wound that tell these skin cells that they need to divide, undergo cell division, give rise to new skin cells, and eventually close up that wound. So that's an example of very successful, very normal cell division. And I think there are 2 points that I want to make about it. The first is that it's tightly regulated. Not only is there a very definite signal that tells these cells when to start dividing, but there's also a very strong signal that tells these cells once the wound is closed, and then they can stop dividing. And the second point I wanted to make is that there is this link that's often described between proliferation of cells and what we call the differentiation of cells. So what you're seeing in this picture on the left are very mature looking adult skin cells. When these cells start to divide to fill up the wound, they actually change a little bit and they become what we call less differentiated. They look less like adult skin cells. They divide, fill up the wound, and then as they stop dividing, they then differentiate into what looks like mature adult skin cells. And I just wanted to make this point here that there's this inverse correlation between proliferation of cells and differentiation, because it's a point that I'll be coming back to later on in my talk. So how is this process of cell division controlled? Well, for a cell to divide, it needs to pass through a series of orderly steps that together form what we call the cell cycle. Now, as you can see here, the cell cycle has 4 major phases. 2 critical steps are the S phase, when all of the DNA is replicated, and the M phase, when the 2 daughter cells break apart and separate. Many of the steps of the transitions from one step of the cell cycle to another are controlled by a group of proteins known as the cyclins and their enzyme partners known as the cyclin-dependent kinases. So what happens in general is that a cyclin protein will bind to a cyclin-dependent kinase, activate that cyclin-dependent kinase, which will ultimately allow it to phosphorylate a range of different substrates and push a cell along into the next phase of the cell cycle. And as you can see, there are different cyclins and different cyclin-dependent kinases that control different parts of this process. In cancer, some of this tight control is lost. And it's obvious and perhaps easy to understand then why we would be very interested in trying to find ways to block the cell cycle in cancer, and in particular to find drugs that inhibit these cyclin-dependent kinases and therefore stop tumor cells from moving from one phase into the next. Turns out that that's much more challenging than we might think. You think it would be very easy, we just make a drug, why don't we make a drug that blocks everything here and then we'll stop all cancer cells from dividing. But it's a really complicated process for a few different reasons. The first one is that different types of cells seem to have different dependencies on different cyclin-dependent kinases. So all the CDKs are important in each cell, but in certain cells, some seem to be more important than others. So one extreme example, if you take the most undifferentiated type of cell that exists, an embryonic stem cell, it seems that they really don't care about CDK, for example, 2, 4, and 6 to proliferate. They really are just purely reliant on CDK1. On the other hand, as cells become more and more differentiated, they have evolved to become reliant on very particular cyclin proteins and very particular cyclin-dependent kinase partners. So for example, a mammary epithelial cell in the breast here seems to be reliant primarily on the cyclin D1 protein and its partner kinase CDK4, whereas a stem cell in the bone marrow, for example, would be much more dependent on CDK6. What makes it complicated is that cancer cells are sitting somewhere between undifferentiated and terminally differentiated cells. So it's not always clear what CDKs an individual cancer type might be dependent on. Another problem is that many cancers will have genetic mutations which make certain parts of this cell cycle regulatory machinery less important. And therefore, it's very difficult to find a one-size-fits-all cell cycle inhibiting drug. A third problem is a problem that we call redundancy. And what that means is that even though a cell might use a particular CDK to divide, so a mammary epithelial cell likes to use CDK4, for example, we often see that if we inhibit that CDK to try and stop a cell from dividing, that it then starts to use other CDKs to divide, to get around the block. And that makes the development of these drugs challenging as well. So when you think about all these challenges, to me, it's actually quite remarkable that we have any CDK inhibitors that work at all. Now, in breast cancer and in breast tissue, there's particularly strong interest in targeting this pair, the cyclin D1 protein and the cyclin-dependent kinase CDK4. And we already know that we have drugs that target this, the CDK4/6 inhibitors, and they're very effective in the treatment of many breast cancers. So why cyclin D1 in breast? Why is this the protein that matters? Well, it turns out that the mammary gland specifically expresses very high levels of cyclin D1, higher than most other tissues in the body. And we know from this very seminal study done 25 years ago that the mammary gland is very reliant upon cyclin D1 in order for its epithelial cells to proliferate. So in this study, the authors made a mouse that was completely knocked out for the cyclin D1 gene. So this mouse has no cyclin D1 at all. And the thought was that this mouse probably wouldn't be able to survive at all because this is such an important protein. Turns out that cyclin D1 knockout mice can be born, and they actually have a small number of very specific defects. And the most striking of that is a defect in the mammary gland. When these mice become pregnant, the normal epithelial proliferation that happens in a mammary gland just simply doesn't happen, and they're unable to nurse their pups as a result. So this was the first clue that there's something special about cyclin D1 in the mammary epithelium. So the hope was, if it's important in the mammary epithelium, that this protein may also be important in cancers derived from the luminal epithelium. And there are 3 very elegant studies performed in the lab of Dr. Peter Sasinski at the Dana-Farber that show us that this is indeed the case. So this first study, published almost 20 years ago, shows us how important cyclin D1 might be in certain breast cancers. So what they've done here is they've made a transgenic mouse, which is pre-programmed to develop a certain type of breast cancer. And as you see this line dropping, you're seeing the individual mice developing breast cancer. If you then take those exact same mice, but delete the cyclin D1 gene, so they don't have cyclin D1 anymore, they're completely immune from forming cancers, showing how important that protein is for this tumor to form. One of the questions after this study was published, well, we know cyclin D1 is important, but are you sure that it's CDK4, the classical partner for cyclin D1, that's also important here? And so that study has been repeated, the only difference being CDK4 is deleted instead of cyclin D1, and we see very similar results. Now, this doesn't mean that every single breast cancer is dependent on cyclin D1 and CDK4. That's probably not the case, but it certainly gives us a clue that they're very important in luminal epithelial proliferation. The critics of this work said, well, that's all very well and good. Cyclin D1 and CDK4 are needed for breast cancer formation, but when a patient comes into your clinic, his or her tumor is already formed. And what we're really interested in is whether the maintained growth of that tumor is also dependent on these enzymes and proteins. And so in a third study, what these authors did was allowed mice to generate tumors, and only after tumors had formed did they selectively delete the cyclin D1 gene. And even here, you can see you have a big impact on tumor growth. So based on all of this work, there's a strong rationale for wanting to try and target cyclin D1 CDK4 in breast cancer. It's also worth noting that in breast cancers, many of the growth factor signaling pathways that drive breast cancers, be that through the PI3 kinase pathway, sometimes the RAS pathway, and of course, most importantly in many breast cancers through the estrogen receptor, all converge to increase levels of cyclin D1 in breast cancer cells. So it's yet another reason why we think this protein is so important. So based on all of that, we would say, okay, it makes a lot of sense that we should try and get drugs that can inhibit CDK4. But it took a long time to get these drugs because it was hard to find compounds that were selective and potent for CDK4/6. As many of you know, we now have these compounds. So what I'm showing you here is a large number of different drugs that inhibit CDK4 and CDK6. The top three we're all familiar with. They're routinely used in our clinics today: abemaciclib, palbociclib, and ribociclib. But there are many other drugs coming up in development as well. They're all potent inhibitors of CDK4 and 6. They're all somehow a little bit different. They have different potencies for CDK4 and CDK6. Some have other targets as well. They have different side effects when we give them to our patients, and in certain clinical situations are even differentially effective. We don't fully understand how these different chemical structures translate into the differences we see in the clinic, but nonetheless, these are the drugs we have, and these are the ones we're using to study this pathway. So how do CDK4/6 inhibitors actually work? I've told you that CDK4 and 6 are important in moving a cell from the G1 phase of the cell cycle to the S phase. What does that really mean? So if we look here, this is a cell, this is the DNA of that cell. There's a family of transcription factors in cells, cancer cells and normal cells, called the E2F transcription factors. And these normally work to drive the expression of a large catalog of genes that push a cell into S phase. There's this very important tumor suppressor protein called RB, the retinoblastoma protein, which normally can sit on these E2F transcription factors and block them. And if RB is sitting there like this, a cell will be stuck in the G1 phase of the cell cycle and not progressing into S phase. When CDK4 and CDK6 are activated, they phosphorylate the RB protein. That causes the RB protein to change its shape, and it moves away from the E2F transcription factors. These E2F transcription factors are then free to drive the expression of S phase genes, and a cell will move into the next phase of the cell cycle. So you would imagine that if we had a CDK4/6 inhibitor, we could reverse this process, that RB would stay stuck to E2F, and that the cells would not be proliferating. And that's exactly what these drugs have been shown to do. So they do work as advertised, and the main effect of CDK4/6 inhibitors is that they do suppress cancer cell proliferation. So here's an example from a clinical trial, a neoadjuvant trial of patients with ER-positive breast cancer, with biopsies taken before and after a 2-week window of treatment with a single agent drug, abemaciclib. And as you can see, in the vast majority of tumors, you see a big reduction in proliferation as measured by Ki-67 staining. If you look into this a little bit more detailed in the lab, you can see that what this is really reflecting is that cells are getting stuck in the cell cycle, exactly where we would expect them to be getting stuck. They're no longer able to progress into the S phase because of the CDK4/6 inhibitor. So that's all very clean and very straightforward. But what I want to put to you today is that, in fact, the effects of CDK4/6 inhibitors are much more complicated than that for a number of different reasons. The first one is that when I talk about these S phase genes, I talk about them very broadly, but it turns out that there are hundreds of different genes in this catalog, and many of them are responsible for very important functions in cells, modulating not only DNA replication, but also epigenetic remodeling, cellular metabolism, et cetera, et cetera. And when one gives a CDK4/6 inhibitor, a cell gets stuck in G1, yes, but also the levels of many of these downstream proteins go way down. And that can also have very broad implications for the biology of the cell. Secondly, not well appreciated, is that this unphosphorylated RB has many other functions. It doesn't purely bind to E2F. And so a CDK4/6 inhibitor will unmask those effects as well. Thirdly is this inconvenient truth that CDK4 and 6 also phosphorylate other proteins, not just RB. And so the drugs will modulate the effect of those substrates as well. And fourthly, there are many other proliferating cells within a cancer, immune cells, fibroblasts, etc. And all of those are treated with the CDK4/6 inhibitor and may be susceptible to its effects and may be responsible for some of the phenotypes that we see in patients treated with these drugs. So that's a little bit about these drugs, why they should work and how they might be a little bit more complicated than we first thought. What I'd now like to do is take you through 3 different examples from our work, some of which I did whilst I was working overseas in the US, and then some of which we've done here at the Peter Mac, outlining some of these less expected effects of CDK4/6 inhibitors. I'm not going to dwell on the classical effects. I'm not going to show you the results from clinical trials that many of us know showing how effective these drugs can be, but rather I'm going to show you some of these perhaps less canonical effects of the drugs. So the first one relates to this somewhat older paper now, it's going on 4 years, that we published whilst I was working at the Dana-Farber. And in this paper, we showed how CDK4/6 inhibitors can often synergize really well with inhibitors of other oncogenic kinases in cancer cells, in many different types of cancer cells. So what did we find in that work? So if you can imagine, here's a breast cancer cell. We know that many breast cancer cells are driven by a number of these receptor tyrosine kinases, whether it be EGFR, HER2, etc., or other kinases that are just below those, feeding into ultimately activation of mTOR. We also know, though it's again not often mentioned, is that mTOR is also an important driver of progression into S phase. And what we're hoping to do when we give patients drugs that block these kinases, HER2 inhibitors, PI3 kinase inhibitors, etc., is to switch off all this signaling and ultimately also switch off mTOR. In this work that we did, we found that there are often cancers that are resistant to these drugs. So for example, a HER2 positive breast cancer that's resistant to blockade of HER2. And when we looked at those cells, we found that was because the drug was able to switch off this part of the pathway, but it wasn't able to switch off mTOR. And mTOR was still on, making these cells resistant. What we then went on to show was that the reason for this was that there was persistent activity of CDK4 and 6, and unexpectedly, CDK4 and 6 were actually driving the activity of mTOR. So this was not a previously appreciated association between these two pathways, but what we found was that if we then added in the CDK4/6 inhibitor, not only did we inhibit the phosphorylation of RB, as you would expect, but we also shut down activity through mTOR, and that led to cell cycle arrest, and we could restore sensitivity to the original kinase inhibitor upstream. What's more, we actually were able to identify the reason that CDK4/6 activity directly feeds into mTOR by showing that the CDK4 enzyme and Cyclin D directly bind to a tumor suppressor protein called TSC2 and phosphorylate it, and that this in turn then activates mTOR. So in our work, we focused on HER2 positive breast cancer, and we focused on what it means when you combine an inhibitor of HER2 up here with a CDK4/6 inhibitor down here. And in many models of HER2 positive breast cancer that was resistant to HER2 targeted therapy, we demonstrated profound synergy from when you added a CDK4/6 inhibitor into the mix. Importantly, since that work was published, this connection between CDK4 and TSC2 has been reproduced by other labs. Firstly, work done in Drosophila, but also subsequently work done in human cells. So we do believe that this pathway is real. It may be carry different levels of importance in different cancer cells, but it is certainly something that in HER2 positive breast cancer seems to be very relevant. And what's most satisfying for us is that this idea that you could take a resistant HER2 positive breast cancer and overcome resistance with the CDK4/6 inhibitor has gained traction in the clinic. Recently, we saw the publication of the results of a randomized trial showing that in patients with refractory HER2 positive disease, the drug abemaciclib added to their anti-HER2 therapy can improve progression-free survival when compared to the standard treatment, which would be chemotherapy. And there are 2 other trials ongoing looking at this exact principle at the moment, and we're keenly awaiting the results of those as well. What's also really interesting to point out is that although we showed this mainly in the context of HER2 positive breast cancer, it's proven that the broad principle that inhibiting an upstream kinase and CDK4/6 at the same time is a good idea has now been proven true in many types of cancer. And in fact, there are clinical trials going on in several cancer types, lung cancer, melanoma, and others, looking at this broad role for CDK4/6 inhibitors to overcome drug resistance. So that's example one. And I guess just to go back to this slide where I said, you know, CDK4/6 inhibitors are more complicated than we thought, I would say this is an example of where a non-RB substrate of CDK4/6, so this I talked about this TSC2 protein, is important and becomes actually clinically quite relevant. So the second example I wanted to talk about is from a separate project that we also published whilst I was overseas in Boston. Another effect of CDK4/6 inhibitors, also very much unexpected and completely unrelated to the previous one. And that was in this paper showing that CDK4/6 inhibitors in various models of cancer can actually stimulate immune responses and encourage the immune attack against cancers. And I show you this example because this is another concept that's starting to gain some traction in the clinic. So what did we show in that work? Basically, what we found was that CDK4/6 inhibitors can turn the tumor immune microenvironment from one that is somewhat cold to, in other words, there's not much immune activity going on, to one which is warmer, where there seems to be some immune activation. And we demonstrated that that occurs through two primary processes. The first is the direct effect of these drugs on tumor cells, where they increase the presentation of antigen on tumor cells through MHC class 1. And completely distinct from that, there were effects we found of CDK4/6 inhibitors on various types of T lymphocytes, which also caused them to become activated or shifted the balance of different types of T cells in favor of a warmer tumor immune microenvironment. After that work was published, a number of other groups have done similar studies, and we've all converged on this same finding. The obvious sort of thought that stemmed from this work was, well, if a CDK4/6 inhibitor warms up a tumor, maybe we could leverage that effect by now adding in some immunotherapy to try and help treat this breast cancer. And we and many of these other groups have demonstrated in the lab that yes, in certain models, animal models, you see very potent synergy when you treat tumors with a CDK4/6 inhibitor and immunotherapy, in this instance, an anti-PD-L1 antibody. And that has led to a number of clinical trials which are ongoing, again, not only in breast cancer, but in other types of cancer as well, where we think that this pairing may actually even have be more impressive. So I don't have time today to go through the blow-by-blow mechanisms associated with that work, but I just wanted to point out again that it's another example of the unexpected effects of CDK4/6 inhibitors potentially being clinically very important. And in that study, many of these non-canonical effects seem to be important. So one of them was to do with the fact that CDK4/6 inhibitors reduced the levels of a particular enzyme, which is an S-phase gene called DNA methyltransferase 1. And that was partly responsible for the effects we saw. Other groups have shown that some of the effects in T cells are related again to one of these non-RB substrates in lymphocytes, a transcription factor called NFAT. And of course, the third is this general principle that these drugs, again, are not only acting on tumor cells, but are also acting on T cells as well. So very complicated in a way, but ultimately converging to a single phenotype, which could be clinically very, very important. Also, just on this point of immunogenicity, I wanted to briefly discuss in one slide a drug that's being developed, a different CDK4/6 inhibitor. This is a drug called trilaciclib. Some of us are familiar with this, but it's probably one that many people haven't heard of yet. This is just another CDK4/6 inhibitor, but what's interesting about this drug is that the company that's making it is developing it with a very different focus. They're not developing this as a drug that will inhibit the proliferation of tumor cells, but rather they're really trying to develop it as an immunomodulatory drug, thinking about the ways that it affects the function of immune cells, be they in the bone marrow or in tumors. It's also given differently. It's an intravenous CDK4/6 inhibitor, and it's given on an intermittent basis, once a week or once a fortnight, something like that. There's been a lot of lab data from this company showing that, like other CDK4/6 inhibitors, trilaciclib can boost anti-tumor immune responses and can dramatically enhance the effects of immune checkpoint inhibition in various models. And it's now a concept that they're really moving to look at into the clinic. The first study that's been done to try and get at this was this randomized trial done in patients with triple negative breast cancer. And here what was done is that patients with this disease, as we know, are normally treated with chemotherapy. These patients received chemotherapy with or without pulsatile doses of trilaciclib given just before each infusion of chemotherapy. And the idea was that the trilaciclib would either boost the anti-tumor immune response in some way, or maybe protect immune cells from the damaging effects of chemotherapy, and therefore make the cancer, put the cancer under better control. It was not designed as a study to stop proliferation of tumor cells. Indeed, this is a cancer where we think these drugs probably won't have a dramatic effect on tumor cells directly anyway. And also the drug, as I mentioned, was being given in a very intermittent fashion. And what was striking about the results of this study, and I think even maybe a little bit surprised the investigators, was that in both arms of this trial, it was a 3-arm trial where patients received trilaciclib, there was a very significant prolongation of overall survival. And that's really sort of brought to the forefront this concept that CDK4/6 inhibitors can be used in a different way. We don't know for sure that the results of this trial are driven by an immune mechanism. However, there was this correlative analysis done from blood samples taken from patients on this study. And it was shown that in the 2 groups of patients who received trilaciclib, the cytotoxic T cells in their peripheral blood were, quote unquote, more stimulated, evidenced by production of interferon gamma. I know this isn't a perfect proof of mechanism, but it's certainly very intriguing. And you'll see more data about trilaciclib in the near future. It's now being developed through the I-SPY neoadjuvant clinical trial program, again, as an immunostimulatory agent. And there may be sort of further trials in the metastatic triple negative space that we could look forward to in the future as well. Okay, so now I'd like to turn to the final example. And as Jeff mentioned in the introduction, this is a more recent project. And this is a project that we've done a lot of the work for here at Peter Mac, where we've been very interested in trying to understand yet another effect of CDK4/6 inhibitors, and that is how do these drugs change the epigenome within a breast cancer cell? And this work has really been led by April Watt, who's a PhD student in my lab here at the Peter Mac. And good timing for me to present because it'll be coming out online in Nature Cancer, I think next Tuesday. So why would we be interested in this potential epigenetic remodeling by these drugs? Well, to explain why we were interested in that, I need to go back a step and revisit what CDK4/6 inhibitors do in breast cancer. So I told you already that these drugs are very good at stopping breast cancer cells from dividing. So here we see inhibition of proliferation. What we also know is that if you leave these drugs on cancer cells for a little while, a few days, for example, that you see another effect. Cells start to develop a phenotype that looks like what we call cellular senescence. This is a process that's normally studied outside of cancer in the field of aging. As we age, our tissues seem to fill up with these senescent cells. These are cells that are no longer dividing, and they're also very different. They're very big, they're very flat, and they are biologically very different. They're not quiet by any means, although they're not proliferating, but they're just very different cells. And it's really perhaps not that surprising that a CDK4/6 inhibitor could make cells look like they're senescent, because when you look at senescence more generally outside of cancer, it's basically considered to be a stress response in many cells. And the way that cells stop proliferating and become senescent is that they find their own internal mechanisms to inhibit CDK2 and CDKs 4 and 6. So what we're doing when we put a CDK4/6 inhibitor on a cell is we're basically driving at least half of the senescence program. And probably that's why we're seeing this senescence-like state in our cells. So what's so interesting about that? Well, what really took our eye was the literature that had come out in recent years showing that senescent cells demonstrate very dramatic changes in their epigenome. And this is all work that's not being done in cancer. This is work that's being done in benign fibroblasts. As I say, most senescence work is not done in cancer. But what had been shown by a couple of different labs was that when these benign fibroblasts become senescent, there are thousands of regions in their DNA, in their chromatin, which change from being very closed and compacted to being very open. And we call these regions enhancers. And when these go from being closed to being open, these enhancers in the DNA are being activated. And what enhancers do, this is a picture of an enhancer in the DNA here, is that they're very cleverly able to bind to the promoters of genes, which may not necessarily be immediately next to them, and activate the expression of those genes. And so what these papers in the benign fibroblast literature had shown is that when cells senesce, these new enhancers get activated and drive the expression of thousands of new genes, which are specific to senescence. This is just the data from 2 of those papers. I won't go over it in detail, but just to mention that this data is obtained using a technique called ChIP-seq, chromatin immunoprecipitation followed by DNA sequencing. And what both sets of authors have done is looked for a marker called H3K27 acetylation on the DNA, on the histone, which is a marker of an active enhancer. And here you can see in this study up here and in this study up here, massive activation of thousands of different enhancers in these cells as they become senescent. So the question which we had was, well, if that's what happens to the epigenome in a benign fibroblast, do you see something similar when a cancer cell is made senescent-like through treatment with a CDK4/6 inhibitor? It's not necessarily intuitive that the same changes would be seen. So we looked at it. And the short version of this story is that many of the same epigenetic changes that we see in benign senescent fibroblasts are recapitulated in cancer cells treated with a CDK4/6 inhibitor. So what I'm showing you here in this first 4 red columns is exactly what I showed you before. This is breast cancer cells treated in a dish with a CDK4/6 inhibitor. And what you're seeing are thousands of different regions within the genome where the chromatin has gone from being very closed to being open. And those same regions are now showing strongly this H3K27 acetylation mark. And for that reason, and for many other reasons, which I'm not going to show you the data for today, we believe that these new regions are serving as active, newly activated enhancers in these cancer cells. And as I told you before, enhancers work by looping around and binding to promoters of genes in the vicinity. And we've actually been able to use some special techniques in the lab to demonstrate exactly that when you treat breast cancer cells with a CDK4/6 inhibitor, you can see these activated connections between enhancers and promoters forming in breast cancer cells. We also see very similar patterns of enhancer activation in our mouse models. So here, these 3 columns on the right, it's the same protocol, the ChIP-seq that I described before, from tumors treated with a CDK4/6 inhibitor. And most strikingly, we've seen very similar things in clinical samples. So on the right here in orange, you're seeing the same thing, the ChIP-seq for enhancer marks in biopsies from a patient on a clinical trial taken before and 2 weeks after a course of CDK4/6 inhibitor treatment. And when you look at the activated enhancers in that patient's tumor and you map them back to the regions in the DNA where we see enhancer activation in cells, they're very similar. So we think that the process that we're seeing in our cells in a dish is really reflecting what's happening in patients. So what do they do? So far, I've just told you that we're seeing senescence and we're seeing enhancer activation. Well, as I explained before, the reason enhancers are so interesting is because they turn on a lot of genes. And I'm not going to show you, we've done bucket loads of RNA sequencing and chip sequencing and all kinds of things to understand this process. But to summarize what we think is happening in one slide, CDK4/6 inhibitors are activating these enhancers in cancer cells. And many of the genes around these enhancers are being turned on and being upregulated. And what we found so far, I think there's a lot more to learn, is that these genes are controlling a number of discrete biological processes. And they explain these processes are directly related to the effects that the drug, therefore, is having on the cancer. The first process, which I'm not going to describe in great depth, is that a lot of genes which we classically associate with a mature luminal mammary epithelial cell are being upregulated by CDK4/6 inhibitors and with a lot of enhancers around them. And we've seen also in our mouse models this striking histologic differentiation of tumors with a lot of new tubule formation. This is something that's also been reported in patients. A second phenomenon that we've observed is that some of these enhancers are driving the expression of immune genes. So that might also contribute to some of these immune effects that I described with a previous project. And the third process is something called apoptotic evasion. And that's the one that I'm going to focus on for a couple more slides. So what do I mean by apoptotic evasion? Well, I've told you that CDK4/6 inhibitors stop cells from proliferating. What I haven't told you yet, but which we have published on before and again in this paper, is that they also prime cells away from apoptosis. So although they're great in that they make cells stop dividing, they actually make it much harder for these same cancer cells to be killed by other drugs that would normally cause apoptosis. And here's a simple experiment that we've done many times now to show this. So here's a breast cancer cell, and you treat it with a you treat it with a drug called staurosporin, which is basically designed to cause apoptosis in all cells, and you see this very robust apoptotic response. However, if that same cancer cell is pre-treated with a CDK4/6 inhibitor for a number of days, and then staurosporin is applied, that apoptotic response is no longer seen. So the cell is protected in some way. It's actually something similar has been shown recently in a clinical trial. This is another neoadjuvant trial in ER-positive breast cancer called the PALET trial. And in this study, it was shown, based on analyses of paired biopsies, pre and post-treatment, that the addition of a CDK4/6 inhibitor, this time palbociclib, to hormonal therapy, dramatically reduces the expression of indices of apoptosis in tumors. So it's a little bit of a clue that this phenomenon of apoptotic evasion is also relevant to the clinic. So why is this happening? So, when our paper comes out, if you're interested in this topic, please feel free to have a look because we go through it in great depth. But in short, what we have found in a number of different cancer cells is that when a CDK4/6 inhibitor is given to these cells, one of the enhancers that gets activated, these epigenetic regions that gets activated, is this long, what we call super enhancer that directly sits over the gene for this protein called BCL-XL. Many of you would have heard of this. BCL-XL is a protein that works as part of the intrinsic apoptotic pathway that usually is anti-apoptotic. So we see this big enhancer coming up over the BCL-XL gene. And interestingly, these are the biopsies from that patient that I showed you earlier, where we also see activation of the same enhancer. And not surprisingly, that leads to increased expression of the gene for BCL-XL. So we looked in 6 different breast cancer cell lines, and in all 6, we saw increased expression of BCL-XL after treatment associated with this enhancer, and also increased levels of BCL-XL protein in the tumor cells. So the idea that we came up with is that CDK4/6 inhibitors activate this enhancer, therefore increasing BCL-XL, therefore protecting cells from apoptosis. Interestingly, in our mouse models, we also see this increase in BCL-XL expression in tumors after CDK4/6 inhibitor treatment. And the obvious question that stems from this is, can we leverage this somehow? If these cells are now relying on BCL-XL to avoid apoptosis, what would happen if we inhibited BCL-XL with a drug? Could we suddenly turn this senescence response into apoptosis? And actually, in our in vivo models and in vitro, which I'm not showing here, that's what we've seen. So if we take this patient-derived xenograft model, for example, we find that pre-treatment of the tumor with a CDK4/6 inhibitor, followed by a short course of BCL-XL inhibition, leads to an increase in tumor cell apoptosis. So it's a strategy that might be in the future, clinically tractable. And it's also really interesting to me because I think it draws a link between cancer and the older literature in senescence. So it's been known outside of cancer that drugs like this, BCL-XL inhibitors and the family, inhibitors of this family, can kill senescent cells. That's been shown before, both in benign cells and also this work from Jeff's lab showing very recently that inhibition of a different member of this protein family, BCL2, could also have very similar effects. So the last couple of minutes, I just want to talk about why is it that we think that these enhancers are actually being activated. So, you know, I've shown you these enhancers getting activated and they could do all these really interesting things. It's a long story and don't even expect you to really read this whole chart, but this is what we think is happening. This is our working model. And if you take away one line from it, we think that the activity of these enhancers is being driven by this family of transcription factors known as the AP1 factors. So these are proteins that many of you may have heard of. AP1 factors typically are the Jun proteins and the Phos proteins, which form homo or heterodimers. There are others in the family as well. And what we have basically shown in this work is that in breast cancer cells, CDK4/6 inhibitors dramatically increase the levels of these AP1 factors in the nuclei of these cells. And this is a process that begins very soon after the drugs are applied and builds and builds and builds over time. What we then also see is that when you get these very high levels of these AP1 factors, you start to see these this is another type of ChIP-seq experiment, you start to see these AP1 factors binding to these regions of the DNA, to these new enhancers that we see getting activated. And we believe that it is this binding of these AP1 factors to these regions, which is sort of it's the transcriptional activity from this binding that is then activating these enhancers and driving the whole process. I don't have time to show you today, but I can just point out that there is old literature going back more than 20 years, which actually provides a mechanistic basis for why it would be that a CDK4/6 inhibitor would increase the levels of AP1. And I'm happy to talk about that more in the questions. What is worth mentioning, however, is that we see the exact same thing in a clinical trial. So this is a relatively large neoadjuvant trial, the NeoPALANA trial using palbociclib. And the investigators in this trial took many biopsies throughout the course of treatment and did gene expression profiling. And what we can see is 4 different AP1 factors, Jun, Jun-B, Phos, and Phos-B. What we can see is that between the first and second biopsies, where only hormonal therapy is given, there's not really any change in the expression of those factors. But then, after a several month-long course of CDK4/6 inhibitors is added, dramatic increase in the expression of many of these factors. Moreover, if you then give a drug, there are drugs available which are sort of very broadly designed to inhibit the activity of this whole AP1 family, we find that many of the gene expression profiles that are turned on by these new enhancers you can prevent that turning on. So here's, for example, this process of luminal differentiation, which I described earlier. And you can see in this green line that this gene expression profile, this luminal profile, is being turned on by abema, the CDK4/6 inhibitor. But if we add the drug here that inhibits AP1 activity broadly, we can prevent that process from happening. So it's another piece of evidence that AP1 may well be involved. And so to summarize this work, I would say that, you know, firstly, we believe that AP1 driven epigenetic remodeling is underlying some of these key biological effects of CDK4/6 inhibitors, and that it might be the beginnings of a mechanistic link, as I mentioned at the start of my talk, between cell cycle arrest and differentiation. A hypophosphorylation of RB, blocking E2F and cell cycle, but also activating a second process which drives differentiation. We're still working out in our heads and in further work how this really might impact clinical practice, the effects on treatment response and drug resistance, and I'm sure other factors and chromatin modifiers are also likely to be involved, and we're starting to look at that. What's very cool is that just a few weeks ago, maybe a couple of months ago, this paper came out when ours was already sort of in press in Nature Cell Biology. Again, nothing to do with cancer, but again, looking at senescence in a completely different context and implicating very strongly that the AP1 factors are indeed very important in this biological process. So finally, I'll just remind you, as I'm going back to this slide, as I like to do, that this is yet another example of where a somewhat unexpected effect of CDK4/6 inhibitors could be very important. And here we're looking at a novel function of unphosphorylated RB. So thank you for that. So to summarize my talk today, I think those of us who work in this field are pretty convinced that CDK4/6 inhibitors are here to stay, at least for the foreseeable future. And I hope that I've shown you today that although these drugs were primarily designed to stop cancer cells from dividing, they clearly do a lot more. I think it's also, just to summarize then, worth mentioning again, that if we're going to develop rational combinations, drugs that we can add to CDK4/6 inhibitors to make them work better, or if we're going to overcome resistance to CDK4/6 inhibitors, in other words, understand why they stop working, we're going to need to understand some of these less classical effects in more detail, whether it's their effects on other cell types, on non-RB substrates, on the epigenome, and other things which we've yet to look into. So with that, I'd like to thank you very much. I'll finish up. First of all, just to briefly mention that I'd like to thank the members of my lab. It's a relatively new lab. We only started up last year, and most of our existence has been in COVID times. So I'm really very proud of what this team has been able to achieve in the great difficulties that they've faced. One of them still hasn't even made it to Melbourne, and Antonio's working from abroad. One person flew here on an emergency flight from Europe. That was Catherine, and spent her first days in Melbourne in quarantine. So really, thank you to my team. Thank you to all the new collaborators that I've been privileged to work with since I joined Peter Mac. Also in other parts of Australia as well. To my ongoing collaborators at the Dana-Farber, to the consumers that have been working with our team, and also for these organizations for their generous support funding our work. Thank you very much.
Thanks very much, Shom. Not sure. Yep, that was a real tour de force. And I think what was so impressive was your presentation, the clarity of your presentation, as well as the science, which is fantastic. So we do have a few questions that are beginning to appear. I might just start, and so please ask your questions in the Q&A section, and I'll read them out. I might ask, because you've got sort of so many elegant stories there, but I'll start with the enhancer induction that you showed through H3K27 acetylation. So obviously BCL2 came up as a hallmark gene. Do you also see MCL1 or BCL2 coming up? And to what extent is that cell type specific for different tumor types?
Yeah, that's a really great question. So the way we got at apoptosis originally was when we did some unbiased ontologic analysis of all the enhancer regions that came up. And one of the terms that was consistently near the top was inhibition of apoptosis through the intrinsic apoptotic pathway. So that's how we got into this, and we knew that senescence is an anti-apoptotic state. So then the next thing we did was we looked at the expression level by mRNA of the classical anti-apoptotic members, BCL2, XL, W, MCL1. And what we found was that 2 of those were consistently upregulated in pretty much all the luminal cells. That was BCL-XL and MCL1. BCL2 was variable. We saw upregulation in some cell lines and not in others. But the reason we focused on BCL-XL and not MCL1 was because Tony Latai, who we were working with at the time, very importantly warned me not to rely on the gene expression data, and said that you must look at the protein levels because many of these are regulated post-transcriptionally. And sure enough, we found that it was only the protein level of BCL-XL and not MCL1 that was going up, which is why we focused on that. We've really confined our studies to luminal breast cancer at the moment. So that's, I guess, in terms of lineage specificity, I don't know, but it's very possible that in other cancer types or other cell types that different members could be important.
So you clearly have some Jeff Lindeman and Jane Bisveda groupies here. The first question is from Jane. For the biopsy subject to ChIP-seq, were cancer cells and stroma immune cells separated if possible? And did you see changes in both?
Yeah, so hi Jane. We in that particular example, we did not. So what we were able to do, it's actually very difficult to procure paired biopsies, as you might imagine, pre and post any drug treatment. We were very fortunate to work with colleagues at the Dana-Farber who were running a neoadjuvant clinical trial where these biopsies were being collected and frozen tissue was being collected. So there weren't many available, but what we were basically looking for was paired biopsies where the tumor cellularity that we could see under the microscope was similar in the pre and post treatment biopsies. So in that particular example, I think it was about 70% in both. And from there, we just went on to get chromatin from the frozen tissue and do ChIP-seq on the whole thing. It would certainly be very interesting, either at a minimum to macro dissect out tumor from those blocks and look again, or even more interestingly, to do something like single cell ATAC on nuclei from the frozen tissue.
So I might go back to your first story where you, I guess, talked about the convergence of signaling onto mTOR and the role that the CDK4/6 inhibitors play on phosphorylation of TSC2. So in your work or in that study that was reported by Fabrice and others, does that become a mechanism of resistance for tumours that progress? So loss of TSC2?
That's a good question. So if a tumor was resistant to combined CDK4/6 plus other kinases.
Yeah, did they have an intrinsic mutation or was that an acquired, one of the mechanisms of acquired resistance, I guess?
Yeah, I mean, it's something that because as Jeff, obviously, you know, there are deletions and mutations that can inactivate TSC2. And it's a theoretical escape mechanism, I guess. I think I've also seen some unpublished CRISPR screens suggesting that TSC2 might be involved in resistance to CDK4/6 inhibitors more broadly. Specifically to that combination, though, I don't think it's been looked at all. So it would just be speculation at this point. But I think it's very possible that that could be a resistance mechanism. It would make sense.
Okay, so one of the attendees has asked anonymously, hi Shom, do you think SASP, senescence-associated secretory phenotype, plays a role in activating the immune system on CDK4/6 inhibition?
Yeah, thanks for that. That's a great question. So the answer is, I think it probably does, but I don't know how. So the SASP is this process where when a cell becomes senescent, it starts to secrete hundreds of different cytokines and proteins. It's called the senescence-associated secretory phenotype. And previous work has shown that outside of cancer, this can lead to immune clearance of senescent cells, often by the innate immune system. So an obvious question is, does that mean that these tumor cells can now be cleared by the innate immune system once you've made them senescent? I think that we're still trying to work it out. There are actually two sides to the SASP. If you read the literature in depth, there is that side where the SASP can encourage immune clearance of senescent cells. There's also another side where it can lead to the production of inflammatory cytokines that actually dampen the anti-tumor immune response. So we're not sure exactly which one of those might be important here. The other point to remember is that most of what we know about the SASP comes from studies of fibroblasts. So for example, IL-6 is the canonical SASP gene because fibroblasts always make a lot of IL-6 when they senesce. Very little has been looked at with regards to the SASP in mammary epithelium. And I believe these cells do have a SASP. They're releasing a lot of different factors, but they're not releasing IL-6, for example. It's just not one of those mammary specific SASP genes. So we also need to dig deeper into what the specific SASP factors are in the mammary epithelium before we can understand that in greater depth.
So I might ask, tell us a little bit about trilaciclib and I guess its potential promise as an IO drug. Does it have preference for CDK4 or 6? And I guess it raises the more general question about the potency of the different CDK4/6 inhibitors as to whether or not 4 or 6 might be more important in immune cells to enable this augmented immune effect.
So that's a really important point, actually. So in broad terms, we think that the more important CDK in the hematopoietic system is CDK6. So some of those studies I showed have actually specifically shown that the effects of these drugs on T cells are driven through CDK6 and not CDK4. It's not saying that there's no chance at all that CDK4 couldn't play a role. With respect to trilaciclib and its relative potency on CDK4 and 6, to be honest, Jeff, I would need to check. I know it's a very potent inhibitor of both, but the relative potency of CDK4 to CDK6, I can't recall.
So do you think abemaciclib is going to be a better drug for immune effects than the other two? Or?
Yeah, so one of the things that the manufacturers of abemaciclib have often said is that they think they hit CDK4 more than they do hit CDK6, and that's why you see, for example, less neutropenia. I actually think that the jury's still out on whether that difference that's observed is clinically relevant. These drugs are all super potent inhibitors of both, and we don't really know yet whether the differences seen actually correlate to different levels of inhibition of these enzymes inside a cell. So I think that we still don't know. Certainly in some of the studies on immunology, people have actually done comparisons. So there are studies where people have treated T cells with abema, ribo, trila, palbo, and just looked at activation in vitro. They all perform somewhat similarly. Perhaps trila and abema have often been a little bit better.
Okay. I'm conscious of the time here and that we've reached time. But I'd really like to thank you, Shom. You've had great numbers attending this forum, and it was a really excellent talk.
Thanks for having me.
And thank you all for tuning in, and hopefully you can tune in again in a couple of weeks' time. Thanks, Shom.
Thank you. Bye, everyone.